Welcome back, everyone, to day two. Folks are seeing a lot of familiar faces and want to keep talking, and as do I, but just for the sake of time and staying on schedule, let's get started with day two of the Upper Columbia Salmon Recovery Award Science Conference. Before I do a few housekeeping items real quick, I just want to, in addition to thanking our platinum, gold, and silver sponsors, I also want to thank Triangle Associates, who we contracted with, and specifically who's now with RCO. They did a lot of coordination behind scenes, so thanks to them. And also, I just want to thank UCSRB staff, specifically Amanda Ward and Gabby Vermeer, who did a lot of behind the scenes work. So thanks to those folks. Just want to make sure they got recognized. Okay, so just a few, I'm just going to do abbreviated housekeeping items. As with yesterday, bathrooms are back there. As with yesterday as well, we'll have these sessions with 4 to 5 talks. If folks get done early enough, we can have time for questions, but we'll have that 15-minute Q&A at the end. For speakers, and then during the Q&A, as with yesterday, we have the standing mic, and then we have the roaming mic, who will be back there. Just raise your hand if you want to have someone bring the mic to you during Q&A. And then just for speakers, just a reminder, at 5 minutes, I'll show this and kind of flash it at you, give me a little nod or eye contact, and then the one minute is here. I'll flash that, and then after that, I'll just stand up here awkwardly until you give me the mic. Anyway, with that, I forgot Janine, 1 sec. With that, let's get started. I want to introduce Janine Castro with U.S. Fish and Wildlife Service. Really excited for her talk. It's on reaching beyond the reach, using a stepping stone approach to restore connectivity. So, thanks, Janine.
So 30 years ago, I was told that I had to work at the site scale, that I couldn't look upstream, I couldn't look downstream, because that was a different property. I was off of the right-of-way. And as a fairly new geomorphologist, just out of grad school, I had to politely explain to my supervisor that he just told me I couldn't do my job. So working at the reach scale to me was like this dream that I had, working at a larger scale. And we've done that. We've worked in large parts of the landscape, upstream and downstream. And so it really struck me that 30 years ago, what I thought would be like the best scenario, working on these really large projects, we're there. We're doing that work. So then I always ask myself, what's next? What's the next thing that we're going to do? And that's why I wanted to talk about kind of reaching beyond the reach. And to do that, I want to take us back in time, and this is McCoy Creek in Oregon. And McCoy Creek suffered the same fate as so many streams in the arid West. It was channelized in the 40s, 50s, and 60s. It didn't recover, but it was a really nice meadow area. And we had this incredible opportunity to restore this meadow. I was working for the Natural Resources Conservation Service at the time, the Wetland Reserve Program, and we had a landowner who was interested a couple miles upstream. So one of the first engineered log jams, kind of big projects I got to work on was actually in the upper part. So McCoy Creek is a tributary to Meadow Creek, which is a tributary to the Grand Ronde River in northeast Oregon. And I'm using this as an example because I think it just kind of shows the evolution of our restoration projects through time. So it's way in the upper part of the Grand Ronde, and there are a number of projects there, and there's some folks here in the audience who've worked on a lot of projects in that area. So if we go back and look at McCoy Creek, one of the first projects I worked on was reconnecting a historical channel. And so we got to build this engineered log jam. It was so exciting. Oh my gosh, we did all these calculations. We were really nervous, like we're putting all this wood in, what might happen? And so it's been monitored over time. So the yellow blob, that's where the log jam is. essentially to kind of shunt water into a historical channel. So if we look at that, we can kind of see how this channel evolved, and it was really exciting. But I would still say this was kind of a site scale project because we were just kind of building the engineered logjam. But if we look kind of broader, if we look at the reach and the opportunity here, the part I just showed you was on the left, so that was the part that was reconnected. But the creek downstream, it was in a ditch, and the historical channels had all been filled in and obliterated. So this was the opportunity. We got to do channel reconstruction. So this is my first big channel reconstruction project that other folks, lots of other folks worked on this through NRCS, Forest Service, Confederated Tribes of the Umatilla, Oregon Department of Fish and Wildlife. There was a whole suite of people who worked on this. Lots of design work to create a single thread meandering channel. And for those of you who are familiar with the Roskin classification, this would have been a C4 channel that held a 1.2-year event. It was great. Like, this was the pinnacle. So we got to do this project. And then we discovered over time that maybe it didn't meet all of our goals. And so you'll just see, as we go through this evolution of McCoy Creek, we started with the single thread channel, like, that didn't quite meet our goals. So some large wood was added, and then some side channels, and there were beaver, and they put in exclosures, tried all kinds of different approaches. And now there's the discussion of, well, maybe we can do a multi-thread channel here and take more of a stage 0 approach. So full floodplain reconnection. And I think a lot of folks would kind of criticize this project as, well, it didn't work the first time. But as a community of practitioners, we're out there on the landscape and we're learning so much. And this was an opportunity early on to try some of these techniques, to see what worked, to do better, do more. But it's not just McCoy Creek. I mean, we've seen these kind of projects all over the landscape. So if we take another step back and we look at where McCoy Meadows fits in this broader landscape, we've got this great habitat patch. But we also happen to have a big habitat patch downstream at Bird Track Springs. And this project was just implemented not that long ago, 2019 or 20, I think. And this is a multi-thread channel project, reconnecting floodplain, creating more habitat. And again, it's a couple miles of stream. So it's a fantastic opportunity that we've got this other big habitat patch. And so then I start thinking, well, we've got McCoy Creek, and we've got Bird Track Springs, and if we look at where they are in the landscape, they're 5-6 miles apart. How do we connect them? And I have all kinds of questions for the fish biologist. How far do the fish move? Does it depend on temperature and flows and time of year? But between Bird Track Springs and McCoy Meadows, do we need stepping stones in there for the fish? And so this is something I've really been grappling with, is how do we connect things up? So I really want you to think about our river systems as fragmented habitat. We tend to think about that in terrestrial systems, and I think we kind of maybe don't think that way about fluvial systems because there's water, and the water's flowing, and so we think, well, it's all connected. But it's really not. It's fragmented by dams, by low flows, by temperature barriers. It's all fragmented habitat. So how do we bring it back together? And I came across this really great map, and this is from the Confederated Tribes of the Umatilla Indian Reservation, and Les Naylor shared this with me. And this is just for the tribes of their projects. So in blue, these are restoration projects that they're actively monitoring. Green are projects that are implemented but not being monitored. And then the red are planned projects. When you look at the Grand Ronde watershed, this is a lot of land. And that doesn't include all the partners who are doing projects in other places. So when we take this kind of watershed approach and we look at all these projects, I'm thinking, how do we link them all up? So I think, where am I going with this? Well, what I really want to talk about are stepping stones. And stepping stone models are really common. They've been around a long time, but primarily for terrestrial species. So this paper that I pulled this from Wang is about monkeys in China and how they move through fragmented habitat. But it got me thinking, I think we could use this. So ideally, we would create corridors. We would just have rivers that are complete corridors. We'd have habitat along the whole thing. That'd be fantastic. And you think about how much money and time that would take, it would be incredibly expensive. And that's what these researchers found when they were studying monkeys, is they couldn't connect using corridors because the price tag was so high. So they used stepping stones. And I think we can do the same thing. And we are already creating these stepping stones, but maybe it's been more implicit and not explicit. And the reason I want to be explicit about stepping stones is for this reason. If you have a small project and you're trying to get funding, it may not score very high. It's like, well, that's only a couple acres. Or, oh, that's only 500 feet of channel. But maybe it's in a place that is so critical that we really need that stepping stone. And so that's why I really want to talk about this, because that is so important. There are a couple of other models out there. This one's from the Forest Service I really like. So looking at habitat patches and existing stepping stones, identifying critical gaps, and then you fill that gap. And we would fill that gap through some sort of restoration activity. And then ideally, over time, you would end up with corridors. So I think that's a really great kind of model to think about. But actually, if we go back to the Wang paper, I really like how they've set this out. So the big green boxes, those are stable habitat patches. The green ovals are stepping stones. And those what they're calling natural stepping stones. And so the first thing you do is figure out those critical gaps, and you fill them. And they're saying that you're filling them with artificial stepping stones. I would say we would call this kind of restoration. So we'd implement to kind of create those connections, those stepping stones. And we would create weak linkages between them. Now, over time, as those projects mature, it strengthens the connections. So we're doing more work, maybe we're getting more flow, and that's strengthening the connections between the stepping stones. And then ideally, over time, those stepping stones coalesce into a habitat patch. And as that habitat patch, we start getting synergy, and maybe that patch grows into another really big, stable habitat patch. We've got strong connections, now we have synergy, and we've got better connectivity. So I think this, for me, is kind of lays out the way I was thinking about our projects, but I hadn't really seen it visually. So this really helped me kind of figure out how can we be strategic in the landscape, because so much of our work is opportunistic. Landowners who are willing to work with us. But if we can kind of look at the landscape and say, these are the areas that are really important, maybe we could just put a little effort to try to get those projects in. And still being opportunistic, but having that strategy. And I think many of you are already doing this, but I just want to make sure we're kind of talking about it. So I want to take you down to the estuary. You might think, oh, this is the Upper Columbia Science Conference. Why on earth would we go to the estuary? Well, hopefully this will kind of make sense when we talk about stepping stones. So I am part of the Expert Regional Technical Group for the estuary. So it is a group of us who are ecologists, fish biologists, geomorphologists. And what we do in the estuary is we review proposed and completed restoration projects in the lower 234 kilometers of the river. So this is basically from Bonneville Dam down to the ocean. We review these projects and we provide scientific kind of technical input and review. And the reason we do it is to assess the potential benefit to juvenile salmon, to your fish, because we exist down there to support, we're doing restoration projects to support upper basin juvenile salmon. We're not there for local stocks. We're really interested on the fish that are moving through. And so I think it is important that in the Upper Columbia that we're talking about the estuary, because all the fish are kind of coming through that area. And just to kind of give you some data to support that, we have a pit tag array at Dairy Creek, Sturgeon Lake, just a little bit downstream of the Portland area. And this is the distribution of fish that we picked up with the pit tag array. And this is fantastic because we're just starting to get pit tag arrays in the estuary. And because we're seeing this data, now we have kind of more evidence for why we need more pit tag arrays. So we've got a pit tag array at Steigerwald now, we've got one at Dairy Creek, and we're trying to get more down there so we can see how long the juvenile salmon, when they're coming in, how long they're staying there. But it was pretty exciting to see this data from all over the basin. So the estuary suffered kind of the same fate as so many of the kind of meadow systems up here. This is kind of what it looks like historically with those habitat patches, and this is what it looks like today. We lost probably over 90% of our tidal marsh, our floodplains, our side channels, they're gone. And so we're trying to restore that habitat. Because I think the estuary was often looked at like a pipeline, the fish are just coming down, they're moving through it, and now what we're seeing, they're using that habitat. And so our focus is on restoring that shallow water habitat. But how does this relate to stepping stones and this landscape approach? Well, I want to share a project with you called the Shire, and it's that red dot in Reach H just downstream from Bonneville Dam. So the Shire, if we evaluated it on a site scale, and this is what we used to do with, and we still do with all the projects, we just take a look at the site scale. What's the access for fish? What's the capacity? Is it a sustainable site? How big is it? So at the site scale, we're like, well, this one's not huge, but it's got some nice attributes. So if we take a look at it, there's several channels. It's only like 15 acres, and for us in the estuary, that's not huge when you're comparing it to like Steigerwald, which is 1,200 acres. And when we score it and we kind of look at the attributes, we look at how much area is inundated for how long, and then actually they have to kind of blast a channel and put in a roughen channel because this area is kind of high and dry because of the change of hydrology. So it doesn't actually score very well. But that's why we've taken this landscape perspective. So the ERTIG exists because of a biological opinion on the Columbia, and basically they've been in existence since about 2009. Now I joined about 5 years ago, just when they started looking at this landscape perspective, which is really exciting. So now we've got this landscape scale conceptual model. So this is kind of the model for us for the estuary. The initial condition, we don't have any habitat. So what's the first thing that you do? Well, the first thing that we did is really looked at tributary junctions. These are biological hotspots. We've got water coming in, nutrients and so forth, sediments coming in, and this tends to be an area where fish kind of hang out. So we started looking at those areas. And then we started looking at, well, how far apart are these tributaries? Can we add some stepping stones to start connecting them? And then ideally over time, you would have a more mature system, and that line with the loops is just time, slowing things down. So this is really kind of the goal, is for the fish to be able to come in and hang out for a longer period of time in the estuary, feed and grow before they go out to the ocean. So if we kind of take a step back and we look at the Shire, that's that red blob kind of in the middle, and you look at where it sits in the landscape, it's really important. It's a stepping stone because there's habitat upstream. That green area is a wildlife refuge, and there's habitat, but there's 23 miles, I think, downstream, or 23 acres, I guess, downstream. But it's a long distance between habitat patches. So we've used this approach to really identify these critical projects, and when we score them, scoring them higher because of their landscape position. So it may not be a great project in terms of acreage, in terms of the type of habitat we're able to restore, but from a landscape perspective, it's critical. And so I think as we are working on projects, you identify these areas, you're going for funding, you need to be really explicit about why this project needs to be implemented, because it is a stepping stone to connect these bigger projects. I think this is going to be important because right now, with all the money coming into the basin, people love these big projects. Like, oh, I'm working on a project now, and they're like, oh, Steigerwald was 1,200. I bet we can get 1,300 out of ours. It's like this competition. Maybe, I'm gonna get 2,000 acres. How much can you get? So I think this is great. Big projects are fantastic. But we shouldn't forget about the small ones that connect everything up. So in the Wang paper, they have 5 steps for their stepping stone approach, which I've kind of modified for our use. And I think it works pretty well. So assess the condition, assess the connectivity in the basin that you're working in, and identify those critical gaps. Figure out some stepping stones. And maybe they're really small, but how do you link up habitats? What are the limiting factors in that basin? And then start proposing corridors. Like if we have patches close enough together, maybe we start kind of connecting things up. And we want these really stable connections. And maybe that means we need more water. And then identify where these key stepping stones are to establish these bigger conservation areas. And this is what I was talking about with synergy between these habitats. And then finally, recovering and restoring that fragmented habitat. And I think that is what we are trying to do. So I think it's a great approach, and it may not be kind of earth-shattering news, but I think so much of what we do, we kind of do implicitly. We know why we're doing it, but other folks don't. We need to tell them why we're doing it and why it's important. So I just want to show some data from the estuary of where I think we are doing this. And I think we are at that point because we're about at a critical mass with a lot of projects. We have enough on the ground now that we can start thinking about linking things up. So this is the estuary, and up until 2021, we had 77 projects. I think we're probably over 80 projects now. We've restored over 10% of the recoverable floodplain area. 10%, that's amazing. So we've mapped, I say we, the royal we, have mapped the estuary and looked at how much recoverable floodplain area is out there. So these are areas that are private lands, behind levees and dikes. We didn't include urban areas, so those were excluded. But we figured out how much recoverable floodplain area is there. And we're at 10% now, which I think is impressive. And I think in many of the basins that you guys work in, start looking at how much have you restored and recovered, and tell that story. So you can see that there are lots of different projects of different sizes, but if we look at kind of the data on the types of projects over time, you'll see that early on there was a lot of acquisition. And I know for many of you working in the upper basin, that may not be an option, but maybe a conservation easement. So there was a lot of acquisition, but in the phase that we're in now, we're doing a lot of levee breaching, levee removal, replacing or removing tide gates, and getting this connectivity. And so it's really ramping up. I think we are gaining a lot of momentum. So if we look over time, we kind of start out, we call this kind of our foundational period, where we started developing projects, figuring out how we're doing this. And then we started kind of implementing. And now you can see in this kind of maturing phase, we're doing some really big projects. And we have some on the horizon that were just fantastic. And I spent the first 10 years of my career working for NRCS, working with private landowners, and I know how difficult it can be to kind of bring them on board to do some of this restoration work with the private landowners. But when there are more projects on the ground and they see those projects, I think the comfort level is there, there's more opportunity. There's not as much fear. Because I know many of you in the audience have been working in this industry for 30 years. And I remember when we started putting wood in streams and everyone was like, oh my, the world's gonna end, like everything's gonna flood, we're gonna wipe out houses. It was so just dramatic. And then we kind of went through that phase, and then we got to this phase of maybe we should like have beaver on the landscape. It was the same reaction, like, we can't do that. And now we're seeing these really big projects. So I think it is, people kind of get used to it, they see our projects, we need to make sure it's out there and people can kind of see what we're doing to bring them on board. So if you want to read more about this landscape approach and using landscape ecology, there's a paper by Hood and others, and it's open source, so you can download it. If you can't find it, let me know. I'm happy to share that with you. And so I'm just going to leave you with a thought before we kind of open up for questions. I think that our goal should be out of reach, but not out of sight. So I challenge all of you in the audience to reach beyond the reach. Thanks. I'm going to stand here awkwardly until someone asks a question. And I tried not to move because it's nice and squeaky up here. It's really hard not to move while you're talking.
Thank you for giving me that transition time to get to the microphone. Janine, really interesting, thank you, to think about how we might reframe how we select projects going forward. So this got me thinking about intensity of individual projects, and I'm curious your perspective on low tech versus high tech in those in-between gaps. You know, I think we think a lot about how much we want to lock in a project or how big we need it to be in order to really create that benefit in that location, even if it's small. But what if we're thinking about this a little differently as something that maybe is more temporary, short, something that's lower tech, like I'm just thinking BDAs, you know, in smaller systems. Super easy, you can put something in, and then maybe it doesn't last. But I was looking at that diagram you had about it strengthening and growing, and maybe that's an approach we should be thinking about. So I'm curious your thoughts.
Yeah, that's a great question. And I heard a few people in the audience actually talking about something very similar earlier. And these kind of high-tech, I'm struggling with kind of the high-tech, low-tech terminology. But I think we've got these low-tech kind of process-based approaches with beaver dam analogs and post-assisted log structures and so forth. And I think anytime we can get out there and get a project in the ground, what we need to do is really be open about how long a project might last, what the purpose is. So let me give you an example. In the estuary, our group has really been resistant to large wood placement because there was a thought that if you put in large wood, then you would find fish right at the wood. And the purpose in many of these projects of placing large wood is not to attract fish to that site, but to change the hydraulics of the system, to create low velocity areas, to get more refined sediment to drop out. So in the same way, I think as you're developing and designing projects, being open about, we're going to do beaver dam analogs, we're hoping it'll last through a 5-year period because we're just trying to kind of get this system. Someone actually talked about conditioning a site. Maybe we're conditioning a site for more work in the future. The way I think about it is anytime you can get land wet, that's important. I mean, from a very simplistic kind of view, we really want to get that land wet. And once it's wet and we're growing riparian vegetation and it starts kind of evolving, then we can go in and do another project. And maybe we bring that along. So I think it's a really good way to get some projects on the ground. And Nicole, I really like this idea of, and maybe that's what we can do at a site. It's quick, it's inexpensive, and it really gives us that opportunity to start building those stepping stones. So yeah, that's fantastic. And I'm really glad you were here because that is something I wanted to really talk about is funding for these projects and how we evaluate and maybe thinking about them a little differently.
I think that was a perfect segue. I wondered, when you're looking at a potential stepping stone, what are the criteria for determining if it's a critical stepping stone?
Oh, that is such a good question, because I need you guys to figure this out. What is critical? How far apart? So in the estuary, what we have defaulted to is we know in a tidal cycle that the juvenile salmon can move about 5 kilometers. So we use 5 kilometers to figure out what those critical gaps are. But what about bull trout? So I was talking to one of my colleagues. He said, well, you know, a lot of bull trout move about 1 kilometer, can move up to 1 kilometer at night. I'm like, okay. So does that mean that we should have stepping stones every kilometer? The fish that you're working with, how far do they move? And I heard someone yesterday say, well, we don't really know. And so that's a critical gap in our knowledge, because we do need to know how far apart are they. I think for now, we can just kind of look at the landscape and say, there's a big stretch between these habitat patches, and we need something in the middle. But I would love that to be more quantitative, based on some science. So that is a research question out there. I think that is something that we need to figure out, but that is a fantastic question. I really appreciate it. One more. Anybody? Okay, come on, last question. Make sure you're all awake this morning.
This may not be meaningful to any of the rest of you, but I'm familiar enough with the North Portland neighborhoods and the lakes there that I'm sure were part of the estuary historically. Do you know if the fish ever wander into like Smith and Bybee Lake or any of those places?
Oh boy, have you asked a tricky question. Yes, there are a lot of lakes in the estuary. They are there, geologically it's fascinating why they're there, but they've been there for a long time. And we have this debate about, do the fish use those lakes or not? And basically say, well, we don't have the data because we can't get out there and sample during high flows. And some people say, well, obviously the fish are using it. Other people say, no, the fish get in there and then there's just a lot of predation. So it's kind of an open question. However, Dairy Creek is connecting up Sturgeon Lake to the river. So we are opening those connections. There is more monitoring going on, but it still remains kind of an open question, and there's a lot of debate about whether that's a good thing or a bad thing. Yeah, thanks for the question. And I think I'm being escorted off the stage. Thank you.
I just mentioned to Janine, we'll actually invite her up to the Q&A after this session as well, so we can ask her some more questions. Okay, so this session is Understanding Habitat Use, and to kick us off will be Rebecca Flitcroft from the Forest Service out of the Pacific Northwest Research Station out of Corvallis, Oregon, and her talk is titled, How Can We Approach Stream Restoration That Supports Diversity for Ecosystems, Species, and Life Stages?
Thanks very much. It is a pleasure to be here with you all this morning, and just a really great talk by Janine. Thank you for kicking this off and thinking about how we connect habitats. And I think that's a really great premise as we move into this presentation. So I'm interested in how we think about aquatic habitats, where they are in the system, and how that affords diversity for the expression of life history. And that's really going to be the core theme of this presentation. But I'm going to start with money, because that's where we all want to start, right? I mean, that's something we're all interested in. And I'm interested in this from the perspective of scale. As Janine mentioned, so many of us are working at sites in fairly small scales. This was an effort by Jaeger and Sherrill that came out last year to look at how investment across the entire Columbia River Basin, how does that translate into the number of fishes that we see? And this is fish passage at Bonneville Dam. And they were able to find that if you look at how much has been spent on restoration over time, there does seem to be a relationship between the amount of dollars spent on restoration and the number of fishes that you see at Bonneville Dam. However, they had great difficulty and weren't able to really find a signal for whether investments that afford diversity and habitat for wild fishes actually resulted in an increase in the population of wild fishes. So how can we move from sometimes the big scale, the big picture, can mask some of the patterns and the results that we want to see at some smaller scales? And some of those smaller scales are some of these projects that Janine has been mentioning today that could be stepping stones if they're placed in the right location within a river system. And there's a wide variety of different forms of restoration, and they're all designed to meet some kind of need or gap that's been identified on the landscape. But how effective is restoration? This is, a key question that we have. And thinking about what Todd said yesterday, well, it depends on how effective restoration is. It depends on whether we actually monitor to evaluate the effectiveness of work. It depends on the questions that we're asking. There's a couple of projects that I wanted to just point out that did do some effectiveness monitoring. This is some work by Carlos Polivka, came out in 2020, looking at how juvenile Chinook responded to large river restoration in the Entiat, and found that there was more time spent in restoration sites and more movement into restoration sites than in unrestored areas. And he was able to go a step further and find that there was a population response to the restoration for the fishes, which is really what we're hoping to be able to see. This project was looking at floodplain restoration, and my colleague Brooke Penaluna led a multi-species environmental DNA monitoring program to understand how the community of organisms shifted in their distribution from the single thread channel that you see in the top to the diverse flooded channel that you see in the bottom. And what we found through this work was that while many of the same species were present in the system, they were more broadly distributed using a lot of the microhabitats that were created throughout the floodplain, which could be considered success for this project. So we are doing a lot of work on the ground. We are evaluating and continuing to do these projects and to think about what needs to be done at a site scale, but our populations are still not where we might like them to be. So what are some other things we might want to consider when we think about restoration and habitats, and what might afford resilience to our native species? One of the things is really linked to this concept of connectivity and variability. I think these concepts are inextricably linked, and we'll explore that in a minute. Has how we see the landscape in its currently modified set changed how we can perceive the types of variability and life history expression that our freshwater organisms are capable of? And we'll talk about that in a minute. And then how can we envision restoration that can enhance the capacity of our freshwater systems to support habitat diversity that confers resilience over the long term? So, starting with connectivity and variability. I think we all are familiar with the kind of 4 key component parts of connectivity that we associate it with freshwater systems. Longitudinal connectivity, really well formed ideas about how systems are linked. You know, Vanote doing such a great job of talking about different communities of organisms from the headwaters to the mouth, and the types of restoration projects that are linked to this longitudinal connectivity piece are our passage projects and removal of barrier culverts and such. Vertical connectivity, this is how that surface water can connect with the subsurface. Different projects looking at embeddedness of landscapes and how we can connect to cooler flow and hyperrheic. Lateral connectivity, focusing on floodplains in many regards, so that we can re-engage connection and restore variability, particularly associated with seasonal characteristics to our river systems. And of course, there's temporal connectivity in river systems. This is how the seasonal and predictable patterns of change in terms of temperature and discharge are allowed, are there for our aquatic species to develop local adaptation to, right? So that predictability within the system over time is a key component of freshwater connectivity. So within that framework of freshwater physical connectivity, there's also the complex pattern of food that's available throughout the system that is dynamic in response to changes in seasonal characteristics. And while I'm not going to speak about this because Ryan Belmore is going to give us a great presentation later today on the foodscape, I just want us to remember to keep in mind that within that broader framework of connectivity on the landscape is also the connectedness of food and nutrients. So what does this mean? Well, as we're thinking about connectivity on the landscape, it becomes more nuanced and complicated by the variation that we see on the landscape. This project was completed by Ashley Steele and published in 2017, looking across a few hundred sites in Western Oregon. And at these sites, information about physical habitat, fish, and environmental characteristics was collected. And one of the big take-home messages from this work was that there's a diversity of gradients of condition on our landscape. So a stream in Southern Oregon is not the same as a stream in a different part of the coast, right? And we know this. So we know that there are north-south gradients, there's east-west gradients associated with patterns of precipitation and temperature possibly, but then that patchy nature of underlying lithology that further complicates the landscape, and on top of that, how people use the landscape, further fragmenting and making more complex those underlying river systems. So how have our fishes been able to survive these dynamic conditions over space and time? And it's through variability and diversity in how they are able to take advantage of conditions that might be good in one year and poor the next. This is an example of upriver migration from Winchester Dam on the North Umpqua. This is in southern Oregon. And what I wanted to point out here was that we were looking at how upriver migration and the timing of migration was linked to patterns of temperature and discharge across the species that were identified at this site. This is a run-of-river dam, so we were able to look more directly at those seasonal characteristics here. And what I wanted to point out here is that some species have a relatively narrow window through which we see the expression of upriver migration, such as with coho. And then you have steelhead that appear to be moving upriver all the time, right? I mean, they're always in the river. They have a much broader range of adaptation to a diversity of conditions in this system. And why do we need to see this diversity of conditions? Because the story about adaptation is not in the mean. It's not in the average time for migration. It's in the tails of the distribution. That's where adaptation to future climate conditions is going to lie. It's in the early returning fish and the late returning fish. And we can see that that capacity to adjust to a diversity of environmental conditions can be expressed at a single species level, in this case, looking at coho salmon more broadly. They had a relatively narrow migration timing at Winchester Dam, but their timing at multiple other sites within their range is highly variable, reflecting the conditions that are locally present at those sites. And it is by being able to shift and change in response to the varying mosaic of conditions on the landscape that our fish are able to survive over the long term. And survive they must, because our climate is changing, and it's changing quickly. What I have wondered is whether the adaptive capacity is present within our fishes to be able to respond to the rate of change. And this really interesting paper by Crozier that came out in 2008 demonstrates that that adaptive capacity is certainly still within the genome of our fishes. So while they are not developing new adaptation to environmental conditions, they are able to use the adaptation that is embedded in their genetics to be able to respond to system change if it occurs gradually enough. So in this case, spring Chinook and sockeye salmon were shown to shift their run timing over time to be sooner in response to warmer conditions likely associated with a changing climate and water regulation. So I've been talking about connectivity within river systems and the broad expressed behavior and variation in our native fishes that allows them to take advantage of that shifting mosaic of habitats that we see across space and time. But are we able to really understand what that looks like and what that means on our landscapes? Because we are evaluating our landscapes through the lens of modification. This figure by Joe Ebersol, which goes back to 1997, is a fantastic way to think about diversity on our systems. And in the beginning, when there was a lot of diverse habitat on our landscapes and things were connected across those 4 levels of connectivity that we talked about at the beginning, there was a multitude of pathways through which our native species could survive and be able to be resilient on our landscape. But over time, as humans have modified both flow regimes and floodplain habitats and estuaries, we have constrained the life histories that can be expressed effectively and that can be survived by our fishes on the landscape. And so in this paper, Joe and his co-authors argued for habitat restoration that restores the resilience of systems by increasing the diversity of habitats so that we could see a re-expression of life history diversity. And then the question is, well, if we restore diversity, is it still there? Will we see the re-expression of variation in our fishes? I think it's an important question. And as we think about our landscapes, this is how we usually think about our rivers, these relatively linear networks with varying pathways of connectivity, often impeded and fragmented, both by features like large dams, which are an obvious impediment to movement and migration, both of fishes, but also of sediments and nutrients, but also culverts in headwater areas, levees that reduce floodplain inundation and reduced estuary habitats. If we think about what that landscape historically looked like, these are those diverse pathways that Joe was talking about in his paper, where we could see diverse expression of survival across the landscape in response to not just the predictable patterns of diversity in terms of habitat complexity, but also those stochastic disturbance events like wildfire or the occasional volcanic eruption that may have modified the types of habitats that were available on the landscape. One project that I think is really useful and a great one to look to comes from the Salmon River Estuary, which is a small estuary in northern Oregon. And before restoration of the tidal marshes began in the 90s, the entire system, except for one of the marshes, had been leveed and diked, and there was just one pathway for life history expression for juveniles moving through this system. And that was to move into the estuary, stay long enough to shift osmoregulation so you could go to the ocean, right? That was the pathway. But after the restoration was completed, multiple pathways of use were documented in this system using otolith analysis. So we had a great presentation yesterday about otolith life history, and this is another place that we've been able to use that otolith life history to understand how fish are using estuarine habitats. And so habitats, we could see that some fishes moved in and stayed for up to a year. Other fishes moved into these habitats for a short period of time, moved back upstream, moved downstream. They were using the diverse habitats that were available in this system. So that adaptive capacity to use diverse habitats when they're available was still present within this population, even though we didn't see it before the restoration was completed. We know that salmon take advantage of dynamic conditions. I love this picture of chum on the Skokomish River in Washington during a flood. They aren't using the river much at all, or at least our road is now the river, right? So when there's dynamic conditions available, our fish are really not shy about giving it a try and looking to see what habitats are available. This is a great study that I think demonstrates that we don't know what we don't know about fish life history diversity. This is a study of rainbow trout that was completed in Idaho by Amanda Rosenberger. And this is also a study that's called How to Save Your Postdoc When Your Sites Go Up in Fire, in Smoke, right? So she had been studying this population of fishes to look at maturity and age distributions, and then a fire burned through their sites. Some of the sites just burned. Some of them were burned and reorganized, so that meant that they also had a landslide that came in. And what was interesting was the sexual maturity at different ages that she documented after the fire. She found that young fishes, young males, were becoming reproductively mature as zeros and as ones. And she also found in those places that were most hardest hit, where there was a landslide and a fire, you saw the emergence of mature females at age 0 and age 1. So while there wasn't much difference in the proportional representation of maturity in those two-year-old fish, there was quite a difference in the young fish. She calls it the live fast, die young life history adaptation to fire. But these fishes were responding in a way that no one had seen before, and we couldn't likely have imagined that they would have to this event. So we've been thinking now about how our life history diversity is present on our systems, but how can we move from these theoretical concepts to thinking about whether we can restore more of this to our systems, to afford our fishes the ability to express their variation and survive in the changing landscapes to come. This paper by Waples from 2009 sets up 3 really important components for this. Thinking about those aspects of connectivity that we defined at the beginning. Thinking about restoring floodplains to allow for pathways of seasonal connectivity through the larger river systems. Thinking about how we can help modify flood pulses and high-intensity events as they move through systems with different types of projects that slow down the water. And always remembering to keep those passage barrier projects coming, because one of the biggest ways that our fish express their adaptation is by moving. And by giving them as many areas that they can move into as possible, that's going to be helpful over the long run. So what does that look like? Well, I think many of us have thought about floodplain restoration. These are a couple of floodplain reset or stage 0 restoration sites. These ones are really hitting on 3 of those key aspects of river connectivity: that lateral floodplain connectivity, the vertical connectivity to the hyporeic zone, as well as affording variation at a seasonal scale to habitats as they emerge and disappear under different cycles of flood. So floodplain restoration can hit some of the marks that we might want when we're thinking about system connectivity. And we know that this type of restoration is something that our biota respond to. This project was completed by Jennings and published in 2023, looking at biomass. And while biomass seemed to drop at a square meter scale immediately after the restoration, the vast area of inundated restoration at the valley scale was such that the overall amount of food on this landscape was higher than it had been before the restoration. We've been monitoring this site for several years after, and there's an ongoing shift in the amount of biomass that we're seeing over time, also in response to events like fire. So these types of restorations can be effective when we start to think about food sources and dynamism across systems. I really like this barrier removal project. This comes from the North Coast, and it's called the Salmon Superhighway. And what's really novel about this project was that they inventoried every barrier culvert in the entire system, and then they evaluated how much it would cost to replace each one, and how much upstream opened habitat would be available. And then they were able to strategically identify, from a cost-benefit perspective, where to prioritize their work, trying to make this work shift from being reactionary to proactive in response to ecological considerations of passage and the economic reality of doing restoration. This type of work will always affect longitudinal connectivity and is a key part of temporal and seasonal connectivity in this system, particularly as we think of how water, sediments, and nutrients move from headwater areas down to the estuary and down into lower rivers. It's those sediments that rejuvenate and enhance habitats throughout the river system, and they can be trapped up behind those barrier culverts. If we're interested in making sure our estuaries continue to be resilient in the face of climate change and sea level rise in the future, we need to be able to feed sediments to our salt marshes as well. And we can't feed sediment to our salt marshes if it's stuck behind barrier culverts in the headwaters. So taking that broader perspective can be really helpful. And I think that broader perspective is really captured in a mountains to the sea perspective as we think about restoration. This is the Salmon River Estuary, where I talked about the restoration that had been done in the lower system. But throughout the entire system and offshore in the marine environment are habitat protections that are in place in this system to try to recover the fishes that occupy this relatively small catchment on the North Oregon coast. But the habitat projects and the restoration, as well as the protections that are in place, are able to cover all of these aspects of connectivity as we've defined it for freshwater systems. So, closing thoughts. Our native salmon are adapted to connectivity and variation over space and time. Our current understanding of life history diversity may reflect observations that were formed under Western disturbed environments, including a propensity to see single-thread channels on our landscape, rather than the diverse historic conditions to which our native species developed the adaptation that's present within those systems. And the key piece here is to look beyond the mean, look beyond peak migration times in downriver movement and migration, because the story And the key to adaptation over the long term is found in the tails of the distribution. It's how can the early returning fish and late returning fish help shift patterns of phenology into the future. We need to recover whole ecosystems to support individual species recovery. And I think Janine offered a really great framework for how we think about whole ecosystem approaches, whole river system approaches to restoration. Phenology is likely going to need to change in response to environmental conditions. So the amount of diversity and variability that we can manifest on our rivers in terms of habitat is going to be critical over the long term. And that leads us to restoration that can allow for all of these dimensions of connectivity as critical both for single species conservation and the healthy ecosystems that support them. And I wanted to close with this image. I was doing quite a bit of work in the Willamette River. It's still part of the Columbia River Basin, just saying. So the Willamette River in Central Oregon. And we were envisioning what this system may have looked like before Western settlement, and that's that pre-1800s condition. And then we have the modern condition on the landscape. We've got channelized systems, we've got some fragmentation, there's barrier culverts, we have dams. There's a lot of people in the Willamette now with hard infrastructure. But how do we want to envision the Willamette in the future? And this is part of a larger effort. We're going to have a conference later in this year talking about how to envision our future river. If we want our system to be resilient for the fishes that live here and for our human communities that occupy this space. How can we envision more diversity in our upland areas? How can we think about restoring connectivity to our rivers and restoring some of the diversity in our floodplains so that we can at least provide a diverse template for our native species to be able to live in as we move into the future? So I would ask all of you to think about the watersheds that you're working in. What might they have looked like? You can see this is a cartoon, clearly, of the Willamette system. But what did you see? What was there before? And how might you want to envision this in the future? So thank you very much. So there is time for a couple of questions. If anybody has a question, I could awkwardly stand here like Janine, alone, with no question. Oh, look, a question. Thank you.
That was an excellent talk. Thank you. I'm always looking for kind of the Occam's razor solution for things like planning and effectiveness monitoring so we can spend more money and time doing rather than thinking. So I'm curious if you have any ideas of your own or favorite ways to kind of quickly and simply assess things like variability and on the landscape.
I think it is truly aspirational to take an Occam's razor approach to identifying and plotting restoration approaches, so I commend you for that. I think there is so much knowledge in this room and so many people here with incredible experience that being able to pull together workshops where you have open frameworks for input and ideas, not just from restoration professionals, but from people who've lived in these systems for a long time. That's something that we've done to try to move forward monitoring strategies for complicated restoration projects. And that might be a way to kickstart something to be able to get as much information pulled together as you can as you move forward. I think so often we are caught up in the day-to-day that we lose sight of our vision. And sometimes pausing and thinking about where we want to go can really help us step back and get back into that day-to-day grind, which is often what it takes to get a restoration project finished.
Thank you.
Yeah, thanks very much. No? All right. Thank you very much.
All right, our next speaker is Virginia White with Hinchinbrook Incorporated, and she's going to be talking, her talk title is Salmonid Habitat Utilization and Restoration, Insights from a Multi-Scale Study.
Good morning. I'm Virginia. I'm a biologist with Hinchinbrook Eco Innovations, and I'm really excited to be presenting behind Janine and Rebecca. I think my presentation is going to echo a lot of what they had to say. So in our study, we investigated the utilization of engineered log jams by Juvenile, Steelhead, and Chinook. And through our study, we hope to gain a better understanding of residency times of individuals within the ELJs and within the reach, as well as movement and activity patterns within individual structures and the reach. And we hope this information can then be used to inform best ELJ design principles for salmonid habitat and future restoration projects. We chose the Stormy B reach of the Eniat River for our study site. The site is ideal because there are 11 ELJs noted with the blue pins in relatively close proximity of each other, various designs and structures, and they're on both sides of the river as well. And you can see that the top half of the site, there's more bends in the river and the structures are a little more closely clustered, and the bottom half, they're more in a straight line and further apart as you move down the river. Our study design had two components, the first being pit tag, mark, and recapture sampling events. And for these sampling events, we conducted fish capture via hand netting or snerting. During data collection, we recorded species, fork length, life stage, and then whether the tagging event was mark or recapture. And we were able to do 10 of these hand capture events from July through December. This included 7 weekly sampling events from July to August, and then 3 more sampling events every 6 weeks from September through December. And yes, fishing in the snow in December was very fun and interesting. And the second component of our study design is array recapture. So we installed a pit tag detection array into an ELJ structure in the middle of our reach, and it's on river left. Oh gosh, sorry guys. Holler at me if I don't keep up with the slides. Okay, so then the second portion of our study is array recapture. So we have an array installed into this structure in the middle of the reach. Thank you. With 5 antennas. This structure was ideal because it's roughly in the middle of the reach, so it has the greatest chance of catching individuals that are moving through the reach. And additionally, it is unique in having a natural log jam, or NLJ, across the river where we could hopefully pick up cross-river detections. And we chose to put the antennas in the more densely wooded part of the structure, on the upstream part, where we observed the most fish. And additionally, within this portion of the structure, the antennas were dispersed in areas of different flow, fish cover, and depth. And we were able to collect the array data continuously over almost 5 months. And then also with that antenna that is across the river, it got knocked loose in November during a high flow event, so we eventually moved it to that location J. So both of those antennas have had less time to capture data. All right, and looking at the movement throughout the reach, these maps show a visualization of the recapture movement for Chinook on the left and Steelhead on the right. The thicker arrowed lines represent a higher percentage of the total movement events in comparison to the thinner yellow lines. And the numbers shown at each lettered site represent the number of fish that were marked and then also recaptured at those sites. So you couldn't see those with an arrow, so I just put numbers at the sites. And many steelhead were either too small to tag or too large and agile to catch. So we tagged about 10 times as many Chinook in comparison to steelhead, but we had similar recapture rates for both species. And to summarize what we're looking at here, there are 4 main points of note. First, between both species, most of the recapture events were at the same structure where the fish was tagged, suggesting high structure fidelity. 2, both species showed movement both downstream and upstream from one side of the river to the other, and movement of varied distances. 3, the closest together structures, such as A and B at the very, very top of site, and E and F about like 3/4 towards the top of site, had lots of movement back and forth, and that suggests that fish may treat closely neighboring structures as one. And then 4th, there is more movement between the clustered upstream structures than the further apart downstream structures. So these results suggest that both species have varied and complex movement activity during rearing, and that juveniles may consequently benefit from restored reaches with complex and varied ELJ structures that are closely clustered together. And then one thing to note, you can see that there are more recapture events involved in the middle of site at site H than any other site, and that's because that's where the array is, so we have 5 months of data for that site as opposed to only 10 days of data at the other sites. So in the future, we would love to either have a few more arrays dispersed through this site or at every site if we could, so that we could capture really a much better picture of all the movement that happens over the seasons. All right, and then now looking at the movement data within an individual structure. So this is the site with the array, and the Chinook data is on the left, steelhead on the right, and the thicker pink arrowed lines represent more movement in comparison to the thinner lines, and then the numbers displayed at each antenna location show the total number of detections at that antenna. So again, to summarize everything we see here, first, antenna K was by far the most visited. That's the one closest to the shore, farthest upstream within the structure. It may have been the most popular because it has excellent cover. It's in a deep pool, and it's in an area of lower flow that's good for prolonged periods of rest. Second, there is movement across the river to the natural log jam, and then also from the log jam back to the engineered log jam, in contradiction to the belief that juveniles maybe tend not to cross the river. And then interestingly, if you look at the image on the right, steelhead also had a few more paths back and forth across the river than Chinook. And then there is also movement all throughout the ELJ, showing that juveniles utilize the full width of the structure in their activities. And then 4th, both species moved most often from the antennas on the bank towards the antennas in the center of the river, and then back and forth. This may indicate preference of movement back and forth between the low flow areas near the bank to the higher flow areas in the center of the river. And then the takeaway here is that similarly to the movement data within the full reach, these results demonstrate the complex movement activities that the juveniles exhibit. And then specifically at the structure level, juveniles may benefit from ELJs with varied habitat for the different activities they're going to be doing in the structure. Low flow areas close to the bank for refuge and rest, and high flow areas towards the center of the river for feeding. All right, and then we also looked at residency times within the reach. So the larger graph is Chinook residence time by day, and then the same for steelhead in the smaller graph. If you look at the Chinook graph, most commonly they had a residence time of around 7 to 50 days, while steelhead had no particularly most common residency time. But among both species, there were many individuals that stayed for more than 100 days, and then between both species, 6 individuals that stayed for more than 150 days all the way into January. And these residency times are much longer than we anticipated, given that juveniles are often expected to migrate downstream much earlier in their life history. And these prolonged residency times may also demonstrate that these ELJs have maybe achieved their goals of improving the habitat quality and habitat availability, and influenced juveniles to stay at the ELJs longer in this high quality habitat before beginning their downstream migration. Wrong way. All right, and then lastly, we also looked at the species activity within the array structure over the course of a day, and then also as the seasons change. These graphs show the number of Chinook detections at each antenna for each hour of the day during September on the left, and then December on the right. And the different colors of the different antennas are represented on the graph. And then also in that smaller image of the array structure, the colors match the antenna color on the graph. So in September, the Chinook were detected much more often and somewhat continuously from 6 a.m. to 7 p.m. And in December, activity dropped and shifted more to dawn and dusk hours. And again, antenna K was the most popular in both months, but in December, activity was dispersed more evenly among the antennas. And then additionally, the number of individuals detected from September to December decreased by about half, from 53 individuals being detected to 24. And that suggests that some individuals may have left the reach during that time. And then looking at the same information for steelhead, they showed similar activity times and detection rates proportionally in comparison to Chinook, with a slight preference to morning activity in December in comparison to Chinook. And in September, steelhead preferred antennas N and M in comparison to K. And then similarly to the results of the overall array movement patterns, these results suggest that, again, the juveniles have complex, dynamic, and varied behavior patterns even over the course of a day, and benefit from ELJs with various habitat types and environmental conditions. And then to close, at both the reach and structure scale, juvenile salmonids may prefer restoration projects that create complex and varied habitat to accommodate their complex and varied movement patterns. And additionally, the surprisingly long residency times for some of these individuals further stresses the importance of designing habitat structures that can accommodate juvenile needs beyond the traditional rearing season, and accommodate their needs as they need to adapt as the climate continues to change. And then I think overall, these results also speak to kind of what Tracy was talking about yesterday, how we're learning that there may be more, well, Tracy and Janine and Rebecca, where there may be more varied life history traits among salmon than we're currently aware of, and it's important that we can capture that data and influence restoration efforts to support a broader range of life histories so they can continue to adapt. Thank you. And I think I have time for one question.
Thanks, Virginia. That's fascinating. Were you guys able to collect any other environmental data within the structure, say, or look at flow conditions or velocities within?
Yes, we took flow and depth and something else that I'm forgetting right now. And Stine, my coworker, is actually going to present and give some more information on that right now. So I'll kind of let her speak to that. Yeah.
As Virginia said, the next presenter is Stine Griep. I hope I pronounced that approximately correct. Also with Hinchinbrook Incorporated, and her talk will be is titled "A Matter of Scales: Assessing Juvenile Salmonid Responses to Floodplain Restoration."
Well, good morning, and thank you for the introduction. Stine was just the right pronunciation for my name. Thank you. So I'm not going to talk about ELJs and all the cool stuff that Virginia did. I'm going to talk about juvenile salmonids and how they respond to floodplain restoration. And what we were looking at, or our research objectives, were to assess Chinook salmon fry and par density and habitat associations on the reach scale. So we wanted to know, are there specific habitat types where juvenile Chinook occur in higher densities? We wanted to compare capacities on different scales, so we wanted to know, are there specific habitat types or reaches or rivers that can provide for higher numbers of fish? And we wanted to evaluate if we can estimate density and growth on the reach scale to learn more about survival and productivities of these reaches, which could feed into life cycle models. And to do that, we sampled in 3 different river basins. So we sampled in the Wenatchee subbasin, we sampled in Nason Creek, we sampled the Entiat, and in the Methow subbasin, we sampled the Twisp River. And just to give a short overview of the sampling reaches that we are looking at in Nason Creek, for those who are familiar with these sites, we sampled Nason 2.3, shown here on the upper left, and we sampled lower and upper Oxbow, shown on the right here, and then we had one control floodplain that was not restored, shown here on the lower left, and those are all located in the lower Nason Creek between river mile 2 and 4. And in the Entiat River, we sampled all in the middle part of the river, so we sampled at area B or Stormy B, here shown on the upper left. We sampled area D or gray D, here shown on the lower left, and area F or gray F shown on the lower right as the 3 restored floodplains. And then again, we had one control floodplain, which was shortly below Stormy B, here shown on the upper right. And they are all between river mile 16 and 21. In the Twisp River, we sampled 3 restored floodplains as well. We sampled Horseshoe. I think there's a great poster out there about that project. We sampled the Twisp River floodplain, and we sampled left bank. So Horseshoe is shown on the upper left, Twisp River floodplain is on the upper right, and left bank is on the lower right. And then we had 2 control floodplains in that river, shown here on the lower images. And they were basically all over the river between river mile 1 and 11. So what exactly did we do at all these floodplains? We had weekly sampling events for 2 to 7 weeks, depending on how long those floodplains were inundated. And in each floodplain, we had 10 to 15 random sampling replicates. And also, we defined different habitat types in each floodplains. I'm going to introduce those habitat types on the next slide. And if they occurred at one of those floodplains, we had 5 sampling replicates representing these habitat types. And our sampling areas had a size of 3 by 3 meters, unless we sampled a natural logjam or an engineered logjam. In that case, we would just sample the entire pool that was formed by these logjams and then measure the pool. And at each of these replicates, we fished or captured and recaptured the fish. Then we identified the species and the life stage. We measured the fork length. And each individual that was large enough, we would mark with a visible implant elastomer, which is like a small colored dot that we would give to them. And we had a different color for each habitat type and a different body position for each sampling event. So depending on when we had a recapture, we would know where was the fish marked, in what habitat type, and when was the fish marked. And also we collected some environmental data. We measured the temperature, we measured the flow and the depth of each replicate. We looked at the main substrate at each replicate. Was it silt, was it sand, gravel, or cobbles? And again, we defined these habitat types. We had open water areas, we had vegetated areas, we had engineered log jams or short ELJs, natural log jams, short NLJs, or we had small artificial channels within a floodplain. And all the results that I'm going to show now are all based on just 1 year of data. So we collected all this data last summer. So to find out if there are any habitat associations and if there are habitat types where fish occur in higher densities, we employed Bayesian generalized linear mixed models, or short GLMMs, and fed them with all those parameters that we measured to see how different habitat parameters affect fish densities. And here you can see our results. So for fry, the results are on the left, and for par on the right. And all those parameters that had a positive association to densities are highlighted in blue, a significant positive association, and all those that had a significant negative association are highlighted in red. So what we can see here is that par had higher densities in deeper parts of the floodplain, so par densities increased with depth, basically. For fry and par, both densities decreased with increasing flow. So in other words, they appear to prefer the lower flow areas of the floodplain. We had a negative association with temperature for fry and a positive association with temperature for par, but we believe that that's probably an artifact because we sampled fry earlier in the season than we sampled par, so the water temperature was still lower. For fry, we also had a positive association with silt, so they occurred in higher densities in silty substrate. We also had positive associations with natural log jams and engineered log jams, so those fish occurred in higher densities in these habitat types. We did not see a difference between restored and controlled floodplains, though both of these floodplain types appear to be have a similar quality, but we could see that there was a difference between the river basins or the tributaries, so that densities were highest in Nason Creek, followed by Twist River, compared to the Enyard. And those estimates here for natural log jams and engineered log jams, we use them as a multiplier for calculating a habitat quality index that I'm going to talk about in a couple of slides. I just want you to remember these are the numbers that I'm going to refer to later in my talk. So for our second objective, we wanted to compare capacity on different scales. So we used density-dependent immigration into the habitat, similar to what Carlos Polivka describes in his publication in 2020. So we compared the number of fish that stay in a specific habitat, so our recapture density, to the number of immigrants, so those fish that we mark newly at a sampling event, and then we fit a Beverton hold model to the data. And we learned yesterday in a talk that if you fit a Beverton hold model, you get a parameter that gives you information about the capacity, and by comparing these values, we learned something about the capacity. And here you see the results for restored floodplains on the top and controlled floodplains on the bottom. And we have these results in bold here for you to see. So, and even though those numbers look quite different, you see with the curves, there is no significant capacity difference between restored and controlled floodplains, which we can see because the confidence intervals of these models overlap. So again, the restored floodplains and the controlled floodplains appear to be of a similar quality. On the river basin or tributary scale, here you see the restored floodplains in Nason Creek on the top and the restored floodplains in the Twist River on the bottom. And here you can see that there is a significant difference. So Nason Creek has a significant higher capacity than the Twist River. For the Enyard River, we did not have enough recapture data, so we couldn't fit a model to the data, so we don't know how the Enyard River compares to these capacity estimates. On the habitat scale, we did not have enough recapture data to fit any models. We're hoping to collect more data next year, but we would see some trends. So you can see, or we saw, that the highest capacity appeared to be in natural logjam habitat. We did not have any recaptures in small excavated channels, so this does not appear to be a high capacity habitat type. And Fry had higher recapture densities in vegetated habitat, while Parr had higher recapture densities in engineered logjam habitat. When we look at densities, we wanted to know, can we estimate densities for entire floodplain regions? So what we did, we used our MARC recapture data, and then we used the Schumacher-Eschmeyer method to estimate abundance, and use these abundance estimates to calculate the number of individuals per 100 square meters. And also we calculated this habitat quality index that I was talking about earlier, which was based on the Shannon Habitat Diversity Index, which is an index that basically tells you how diverse is my habitat, how many different habitat types do I have. And then we added those multipliers from the GLMM that significantly affected the density of our juveniles to give those habitat types a higher weight, basically. And again, you can see those that are statistically robust. Those results are highlighted in red here. The others were not statistically robust. But you can see, again, that the highest densities occurred in Nason Creek, and that the densities are in accordance with the habitat quality index that we calculated, at least within the river basin. So more diverse habitat obviously appears to result in higher fish densities. For the Antioch River, we saw that we have high quality in habitat quality indices, at least for some floodplains, but again, we didn't have enough recapture data to estimate the density or the abundance. But given the study that Virginia just presented, where we saw a lot of Parr and a lot of recaptures, we think that the Antioch River probably has a large but open population. We also wanted to know if there were differences in growth, so we calculated the mean fork length for each floodplain during the first sampling event and during the last sampling event, and then we calculated the mean weekly growth rates to compare them, and you can see the results here. So on the x-axis you see the different floodplain reaches, and on the y-axis you see the weekly growth rates, and those little numbers on top of the bars show how many weeks went into our estimation, because some floodplains ran dry faster than others. And what you can see is that the growth rates were highest at Left Bank and Horseshoe in the Twist River, both floodplains that had a comparably low habitat quality index. So we believe that competition might limit growth in some of the floodplains with a high habitat quality index, like, for example, the Twist River floodplain, which was a floodplain where we also saw a lot of yearlings when we sampled. The high growth rates in gray F are consistent with the high habitat quality index that we estimated for the Antioch floodplains. And again, we can see that the growth rates were comparable between the restored and the control floodplains. So to summarize all these results, on the habitat scale, Chinook, Fry, and Parr select habitat on floodplains, which is consistent with previous observations for early life history, but some of the parameters appear to affect those life stages differently. On the reach scale, restored and natural floodplain reaches provide comparable habitat quality, so they appear to be similar. The Chinook densities are highest in floodplain regions with diverse habitats, so we have high densities in diverse floodplains, and the mean weekly growth rates was highest in floodplain regions with lower capacity and lower density, and we hope to be able to collect more data to answer the question why that's the case. On the river basin scale, Nason Creek had the highest capacity, followed by the Twist River, and the Antioch River appears to have a large open population. And with that, I would like to thank all those who funded us, which were the tributary committees and the Pacific Northwest Research Station, and all those that provided information and access about the different floodplains that we sampled, plus all those private landowners, and of course our crew who did all the hard work last summer. Thank you. And there appears to be time for one question.
Was really great presentation and really striking to see, really from glancing at the slides, an almost tenfold difference in densities between Nason Creek and the TWISP. Even as it seemed like the Habitat Diversity Index was predicting within systems well, but not necessarily between systems. Why do you think you'd have that almost tenfold difference in densities? Do you think it's just a function of the site or a real difference in systems? And I'm just thinking in the bigger question, how do we identify those productive hotspots on the landscape and why they are that?
I think one of the answers might be that, especially in Nason Creek, all the floodplains that we were looking at were perennially inundated, so they didn't run dry, while in the Twist River floodplain, which appeared to be very good habitat, ran dry at some point, except for some pools, and the control floodplains ran dry. So, and Horseshoe and Left Bank are mostly large excavated side channels, so they are not as diverse maybe as other floodplains. That would be my feeling about it, but I would love to collect more data about it to give a decent answer to that, I guess.
Okay, last but not least for this session is John Crandall with the Methow Salmon Recovery Foundation. His talk is titled Beyond Salmon, Lamprey Use of Salmonid-Focused Habitat Restoration Projects.
Good morning. It's a pleasure to be here this morning to speak to you, and dare I say we're going to take Todd's advice this morning and change our perspective, shift our focus, and shift gears and mention lampreys in what has by and large been a very salmonid-focused conference to date. And maybe I'm wrong, but to this point, I don't think the word lamprey has been spoken here on this podium. So we're going to move forward, and I think there's some good reasons to do that. And I don't think that we will obtain meaningful salmonid restoration and recovery without also obtaining lamprey recovery. And there are some similarities and differences between these species, between these fishes, that are worth looking at and taking stock of to figure out how we can learn from lamprey and what they can teach us about how we restore our stream ecosystems. And in the current environment, we're spending upwards of $20 million a year in the Upper Columbia to restore through salmonid-focused habitat projects. Compare that to what we're doing and how we're focusing that work with lampreys, and it pales in comparison. So what we tried to do with what I'm going to explain this morning is how we can look at salmonid-focused restoration and to determine whether or not that's having any measurable benefit for lampreys. So they're kind of going along for the ride. And I'd like to thank Bonneville Power provided funding for this study, and we had a really great project team from a number of different groups that are listed up there that participated in this project. And I think we had a good time doing it, and we learned a lot along the way. So I want to share some of that with you this morning. Okay, so similarities and differences between lamprey and salmon, and there are a lot. And if you look at those limiting factors that are identified in the kind of management plan that we have for lampreys in the Upper Columbia, those five look pretty similar to what you would see for salmon. They're very well known to us: low adult abundance, habitat degradation and alteration, that's loss of floodplains, diminished in-stream complexity, passage issues, dams, different things like that, water quality, and then dewatering and flow management. And I think with the dewatering, that's one place that we have a lot to gain from looking at how lamprey respond to that as opposed to salmonids, and I'll touch on that in a little bit. But overall, the limiting factors for lampreys in the upper Columbia and kind of Columbia Basin wide and wherever lamprey are very similar to what we see for salmon. But because the life history of lampreys differs in some significant ways, especially during the larval phase of lamprey, that there are some differences in how those limiting factors are playing out. So, similar to salmon in that low abundance, low adult abundance, these are the counts over Wells Dam for the about past 2 decades, and you can see while there's been a couple pretty happy years in terms of lamprey returns over wells, there's been a fairly significant flat line for a bunch of years. And this has resulted in very low adult abundance returning to, especially above Wells Dam, to both the Methow and the Okanagan, and until very recently, lampreys, Pacific lamprey in the Okanagan were functionally extinct or extirpated from that basin, and they were on their way. If you look at about 2014, 2015, they were getting very difficult to find in the Methow, and also the age structure of that larval population, which is kind of the easiest way to sample in stream. The adults can be difficult, especially at such low abundances. So what the larvae were showing us was that the population was both getting smaller and then also aging. And something changed in 2015 thanks to a tribal-led effort to translocate adults from lower Columbia dams, captures at the lower Columbia dams. Those fish were essentially truck and hauled up to the Methow and the Okanagan a couple years later, and those translocations of adults have significantly increased the adult abundance of fish moving into both of those basins. And it's kind of a different talk, a different presentation, but that translocation effort has been wildly successful in reestablishing both the abundance and then also increasing the distribution of lamprey within both of those basins, both the Okanagan and the Methow. So kind of a different story, but just kind of some context for where we are in terms of the low abundance and why it makes sense, or we should be looking at lamprey as well as salmonids, but it's a different fish, and if we can step back, take a broader perspective of what we're doing, even though we can focus on lamprey as a single species, because of those habitat differences, it's going to allow us to develop more holistic restoration efforts across the upper Columbia and elsewhere. So I mentioned lots of funding going into Salmonid-focused habitat restoration projects. We're all familiar, we're here at a conference kind of looking at fish use and habitat restoration. We're doing a lot of this work, people are very familiar with it. A lot of projects going in, but generally these projects are designed and planned and funded with salmon in mind. And there's a lot of reasons for that. The ESA and having that federal protection for the listed salmonids in the upper Columbia drives funding agencies and others to support and develop projects for all those reasons that are good reasons. Pacific lamprey are not listed, even though we know we have very low adult abundance for a lot of reasons, not always ecological or biological, probably more political. Lampreys have not been listed, so we don't have those protections. So taking a step back and allowing salmon to drive the restoration, and us taking a step back and looking at, well, how can this opportunity that we have through the funding mechanisms and the project planning and the whole salmon recovery mechanism framework, how can we use these projects to help lamprey? And I think one of the take-homes, if I could get out of this talk for you all today, is never approach a salmon restoration project planning meeting without leaving that meeting and mentioning the word lamprey. Say lampreys. Just get good. Say it, lampreys. And just get good and comfortable with it. Eels, whatever that word needs to be for you to get that word out, to bring them in, because it's going to be much easier to incorporate lamprey planning early in project development. And I really want to stress that because there's a lot of benefit. It doesn't necessarily have to increase project costs. It just takes some thought and intention towards bringing them along for the ride into this mechanism. So what we wanted to do with our project is figure out how some of these Salmonid-focused habitat restoration projects were providing habitat for lampreys. And I think this is a great slide, this great image showing how lamprey can latch on to salmon and go for the ride. And if anyone ever tells you that lamprey can't jump, now you can look them straight in the face and say, absolutely they can, and they can climb too. So by using this, they got way more ways to get around an in-stream barrier than salmon do. So pretty impressive, and they are tight. They've been around on this earth for hundreds of millions of years, and they have a lot to teach us. And again, there's a much more holistic way to look at our recovery efforts if we're including salmon in the conversation. One of the main getting at that kind of second factor of limiting factors is habitat degradation, in-stream habitat complexity for salmon, especially juveniles, installation of large wood, connecting floodplains, as we just heard about, seeing those wood structures in the ENIAT earlier with all the fish movement. Those are also potential habitats for lamprey, as we will find out. But restoring in-stream complexity is a very pervasive avenue of habitat restoration in the Upper Columbia and elsewhere. It's super critical, largely for the juveniles in the Upper Columbia. And one of the mechanisms, techniques, tools we're using that is the installation of engineered wood structures, log structures, ELJs, whatever you want to call them. As we have been working on sampling for larval lampreys over the years, we got to see, working in some of these project areas, and we're kind of noticing the development of lamprey habitat for larval rearing down in association with these wood structures. And so there are a few of us were kind of batting around ideas, and then we just well, let's take a look at it. Let's take stock of how these projects are doing with providing lamprey habitat. And so that's one place that there's a significant difference between how rearing lamprey utilize a stream and how rearing salmonids utilize a stream. The wood is put in for cover, for depth, for velocity. Salmon are really good. They're visual feeders. They're darting around, as we just saw. Super interesting to see all that data from those wood structures. Lamprey, if you come up to them, you actually could snorkel all day and not see them because they're buried in the sediments. And so one of the things we wanted to do was at each of these structures classify, have they been have the ability to either develop or maintain larval rearing habitat. And we call this, it's patch-based habitat. It's this, and you can see in the bottom, I guess that'd be right, this is type 1, how it's classified, and that generally consists of fine sand, detritus, and silt, very fine sediments. So all the things that get into the gravel and clog reds, and we found that are terrible for salmon, that is just like the most ideal larval rearing habitat. So this is not only fine sediment, is just a part of our stream ecosystem that moves through it. It's really important. It's not a bad thing. It depends on where and how it shows up in the stream. That could be a negative thing. But for rearing larvae, silt and detritus and fine sand is critical, and you will almost always find the highest densities of larval lampreys in these fine sediments. And when you jump over that black line into what we call type 2 or type 3, type 2 is will be generally suitable, but much less suitable than type 1. It's generally going to have coarser sands and then also kind of gravel and a little bit of cobble. Once it gets above about 50% cobble and gravel, it's type 3, and it's generally unsuitable, and you will very, very rarely find any rearing lampreys in there. So we wanted to visit these structures to see if they were providing this type of habitat, which is rearing habitat. And so this is another interesting thing within a stream environment. This type of substrate is discrete. It forms in discrete patches, largely related to water velocities, also depth. Oftentimes you'll find this downstream of rocks, on the inside of bends, in backwaters. This is supercritical habitat, and there are stepping stones as you move down between these larval habitat patches. There's a whole open door of study about how these fish move between these stepping stones of patches, but we were focusing on all these structures. And so what we did is went out in both the Methow and Entiat subbasins and visited a number of projects that we've been hearing about that have all these wood structures, and we just basically took stock and an inventory of what the wheres and whens of these different structures and tried to determine how much larval habitat was associated with them. And then as kind of an added bonus, were lampreys present in those habitats? So there was kind of a two-phase thing, but really what we were focused on is trying to figure out if the habitat was even available at these structures. So the most coarse way we did this at each project site was just to generally classify the wood structure within the reach. And we put them into 3 categories, just kind of picked numbers, large, small, and simple. So it's kind of more than 10 logs was a large structure, 4 to 10 was a, oh boy, was a small. So moving through, basically this is just all the associated data we took with the wood structures, like to put it into context. Here is a gratuitous table of a ton of numbers you probably don't want to read. And it's probably in too small a font for you to read anyhow, but basically we did a lot of large structures that are put out there. They're generally in the main stem, and they're in glides. And so that is really a report card of where the habitat structures are existing, where we're putting them from all these different projects. So here's a structure, there's a larval habitat patch, that is what was sampled. And so as we go out there, we found that the large structures had the most area of presence of larval habitat patches, but they all had combined over 80% of the wood structures over the 200 structures we went to had some formation of a larval habitat patch. The size of the patch was super similar between the Methow and the Entiat, and then within the different types of the large, smalls, it was generally a pretty even distribution of the patch size in relationship to those types of structures. The percent of type 1, which we know is that really good larval habitat, that was very similar between the Entiat and the Methow, and then in the smalls and the large structures had a little bit more of that really good type 1 compared to the type 2 at those structures. And the densities overall were very similar between the Methow and the Entiat. This is the number of larvae within the patch, and then across the larval densities with the different types of the ELJs. And I'm going to get kicked off here, so I'm just going to blow through here. You can read that big structures were good. We had a lot of velocity information that would show the more intercept of the stream channel with the structure, promotes deposition of fine sediments. No crazy thing to think there. And just remember lampreys. Put in best management practice. This is a salmonid-based project right here. Looks very simple, but it quickly turns into a lamprey project when you isolate all this fine sediment, and I guarantee you that pump right there, screen, is probably sucking up several different year classes of lamprey as it does that, even though we were really well-intentioned to prevent it from pulling up salmon through there. Ditches, screens, and dewatering, it's something that is widespread in the Upper Columbia. We need to be paying way more attention to that as we do this, so we don't end up in situations like this where we're doing triage on ditches. We can do way better than this, and we just need to plan for it and come up with creative ideas and solutions so that we're not annihilating multiple year classes of lampreys as we do this. So we can do better. We're starting to really ramp up our efforts, and that's great to see, but I know we can do better. So thanks. And remember, say lampreys at your next meeting.
All right, I'm going to invite the other speakers from that session, including Janine. So Janine, Rebecca, Virginia, Stine, and then John. We'll put John in the penalty box for 5 minutes. He can't answer a question because he went over. No. So, yep, we got, about 12 minutes or so for questions per the usual arrangement. Standing mic, roaming mic person over there. So raise your hand and don't make me ask a question. I have some, so.
Excellent talks, thank you. I had a question for Virginia, and that was looking at the, well, I kind of have two questions if I'm allowed. I was thinking about a lot of the home range research that had been done when radio telemetry first came out. I mean, it seems like in the 80s and 90s there was a lot of work on the home range of adult and juvenile salmonids, and so I was wondering if you looked at how that compared with what you saw, because I think kind of what I saw, it looked like you were covering maybe a kilometer or so of river, and I think the home range for a lot of juvenile and adult salmonids is 100 meters to a kilometer. So I'm wondering if what you thought of, your work, how that compared with that.
Yeah, so we, our main project last year was the floodplain work that Stine presented on, and we're like just diving into this work, so we haven't really got there yet. But we did that at the snorkeling at Stormy Bee, 2 other restored reaches, and then 3 control unrestored reaches. And just to kind of speak on the movement between reaches that we've seen so far, we have seen very little movement, like greater than kind of that 1 kilometer distance, but there have been 2 individuals who have moved down to the array structure from about like 10-ish miles upriver, but we haven't quite gotten to look into the home range detail.
Thanks. Well, this is probably my second question. You're probably going to, you probably haven't gotten to this either, but given that you pit tagged all these fish, are you going to look at their movement and when they're being detected, either in smolt traps or down through the dams? And maybe they haven't outmigrated yet, since it seems like a fairly recent study.
Yes, I attempted for the first time yesterday to see if we've gotten any downstream detections, and either I did it wrong or we don't have any downstream detections yet, but we are hoping to see them pop up further downstream.
Great, thanks.
Yeah.
I can add to that. I think we had recapture at the small trap, actually, from one of our pit tags down at the end, yeah.
I think this is for Rebecca and Virginia. Have you seen anything to suggest that a resident life history strategy in Omicus is becoming more dominant in restored reaches that you're looking at?
I haven't looked specifically at life history expression in Omicus. I would refer to some of the literature on the topic about cortisol levels as a key part of the shift from resident to anadromous life history in Omicus. So higher cortisol levels, and you have more steelhead. Lower cortisol levels, you have more resident Omicus. So there's some literature on that topic out there. And there's some folks who theorize that we may end up with more resident Omicus because of differences in freshwater habitat in the future, but I don't know that we're seeing that yet.
I guess I think we tagged maybe, I can't remember exactly how many steelhead we tagged, but I think the graph I showed, there was almost perfectly even distribution among the residency times, so we're not really seeing a trend with our small sample size.
So I'm going to say the word lamprey. Basically, I think it's more of a John or Janine question, but I think John really lifted the point about how salmon ride the sort of coattails of, or lamprey ride the coattails of salmon projects, in particular the fairly common engineered log structures. I think most probably consider full floodplain projects and your stage zeros and that sort of thing to be a little bit on the newer side compared to engineered log structures. from what you've seen so far, say of Weich's or Mackenzie, the South Fork Mackenzie or these types, can you just comment on how Lamprey, early results maybe on Lamprey with those types of full floodplain oriented projects?
You go ahead.
I kind of touched on it, but I ran out of time. The one thing I didn't point out is we did our study at low water. So what the numbers that we were coming up with would be for sure an underestimation of habitat that was created or maintained by those wood structures. And the one thing we did come up against is there were a bunch of sites where we saw suitable habitat, but it was dry. So if you went back at high water and sampled, the patches would be larger. Who knows what that did with the densities? And so it's a question I've had, and there's very little information on how Pacific lampreys or probably other larvae move between the patches or within a patch if it's large. But I think given the potential for that type of substrate and seeing some of those results from the Mackenzie where you're getting more dispersal of lampreys across a floodplain, that they will occupy those habitats if they become available. But it is really an interesting thing as stage goes up and down, where the cues are, like, hey, it's time to bail, so they don't end up getting stranded. And we do find larvae stranded in isolated pools in the Methow and elsewhere.
I would just make a comment about the South Fork Mackenzie. So there's a project on the Mackenzie system to try to understand how lamprey are using the stage 0 sites on the South Fork, and there's some other stage 0 sites in that upper portion of the Mackenzie as well. And so they marked quite a few returning adult lamprey in some of the dams lower down in the Mackenzie system. And the vast majority of those fish are homing to the Mackenzie. So we do know that there's adult dispersal moving into those sites. Because the South Fork is below a dam, there is a regulation in terms of flow, so that system doesn't bottom out and dry out quite as much at base flow as you might see in some other places. So some ongoing research is being done by ODFW research to try to understand juvenile lamprey occupancy, but it looks like the stage 0 sites offer an enormous amount of rearing capacity for juvenile lamprey.
Thanks for these talks. It's so refreshing to hear the diversity topic come up and the complexity pieces of this. And as a kind of consultant designing these projects, I think some of the worst projects I've ever had to build have been focused on one life stage of one single species. And the kind of resilience of that habitat is diminished sometimes by that restoration project. It will work if all other things are working in that system. So I'm wondering, you did mention this connection between funding and the success of restoration. I'm wondering if the funding, are you seeing a shift in the funding of these projects that allows for more complexity and connectivity versus this focus on this kind of myopic single stage, single species approach?
I think, thanks Marjorie for that question. I think there has been a shift in funding, and certainly from the Fish and Wildlife Service perspective, the money that we're getting in, we're really looking at bull trout, lamprey, mussels, some of these other species, and looking more broadly at life stages. So I think there has been a shift towards multi-species, more complex. So I'm hoping that pattern continues, and it does seem like we're moving away from that really single species focus. And because I think a lot of the projects that we're now evaluating for funding, like how many species can we actually address through this project? And that's definitely a beneficial aspect. So I don't know if anybody else has comments on funding.
Yeah, I would agree in the, I sit on a few restoration recovery boards as well, and broadening the scope to be more inclusive definitely seems to be the trend that folks are going on. because we are living in a single species conservation environment still through ESA, of course, making sure that we can include habitats for our specific target species, but trying to see how we can leverage that to affect a broader array of species seems to be what I'm seeing in both the design of projects and in the funding mechanisms right now.
Thank you, Ryan. So one of the things that I've been thinking about in the relationship to David's thing about social sciences and storytelling is, Virginia mentioned she wasn't sure if she'd used the database to find the pit tags. And I know that there are birding sites and I think butterfly sites. And in the bird cases, people can look up if they spot a bird with a tag and it ends up somewhere else, they can find out where did that bird go. And those are publicly accessible databases. And I've supported Salmon in the Classroom for 20-something years, and the children don't get to just go look where their salmon ended up, but it has to come through the gatekeeper from the conservation district, finding out that some of their salmon went through Bonneville, and reaching out to the teachers, and if the teachers notice it in their email and bother to tell the students. So can we figure out a way to get publicly accessible movement data that's very easy for our kids who are participating in these programs, or for our young field staff, researchers who are engaged in injecting these pit tags, so that if they move on and they're in grad school 2 years later, they can see if any fish they handled are still alive. And where do they go?
I love that idea. I think it's fantastic. I would offer that the Columbia Basin is far ahead of much of the rest of the West Coast in terms of having a centralized repository for pit tag information, which is pretty impressive. I'm working with the Confederated Tribes of the Umatilla, trying to understand where their lamprey are going. So, we can access the database for the Columbia River Basin pretty well for the pit tagged lamprey that they are sending out into the universe, but it has been it has been challenging to get information from other places on the West Coast so that we can see if Columbia Basin lamprey are showing up elsewhere, because they don't seem to be returning in the same way that salmon do. So, I think there's a broader opportunity with something like that to create a West Coast wide, including Canada, in that for pit tag information, some kind of a standardized base, because right now it's not standard.
Yeah, and I would agree. I think that's a really fantastic idea. And there is a database, and the problem is we've got pit tag arrays that are being managed by a whole variety of different groups, and we're trying to bring that data together. But I think the next step is that communication piece. Like, how do we take that data and share it more broadly with the public? And we're not really doing that yet. So if you can get in the database, you can find specific things, but communicating back to the teachers and the students and so forth, I think that would be the next step. We need a better kind of user interface for that, because we've got the data, but now how do we share that? And I think that's definitely the next step.
Let's thank our speakers. So break is 10:30 to 11. I just want to mention that the posters are still up. I think most of them are, so if you didn't have a chance to check them out, go ahead and take a look. Thanks. See you back at 11.